Yeah. Okay. Yeah. So today we are going to learn about human health and diseases. Second chapter, strategies for enhancement in food production, then microbes in human welfare, and the next chapter is the ecosystem. Okay, sir? Okay. So, the first thing we have to learn about is the immunity. Immunity refers to ability of the body to protect against all types of foreign bodies like bacteria, virus and foreign substances like toxins. In very simple definition, resistance against the diseases is called as immunity. So there are some important characteristics for immune system in our body, that is it should be able to differentiate self from the non-self. Okay, and there are three features of immunity system: recognize, respond and remember. This we call it as the 3 R's. First, the body immunity system should be able to recognize the foreign bodies that enter, then it should respond to that and it should remember about the antigens so that it can protect in the next attack. Then immunity system mainly consists of lymphatic organs, tissues, cells, vessels and also lymph. Now we learn about the types of lymphoid organs. There are two types of lymphoid organs, primary lymphoid organs and secondary lymphoid organs. Okay, in the primary lymphoid organs, production and maturation of the lymphocytes takes place, whereas in secondary lymphoid organs, proliferation and differentiation takes place. So, bone marrow and thymus are the primary lymphoid organs. Bone marrow is the center for stem cells of all types of cells, including the lymphocytes. So, here B lymphocytes and T lymphocytes, both of them will be produced, but maturation of the B lymphocytes occurs in bone marrow itself, whereas maturation of the T lymphocytes occurs in the thymus. And all of you know thymus is a bilobed organ which is present near the heart, and it is very large during the birth. As we ages, it shrinks. Now, secondary lymphoid organs I already discussed, means I already mentioned proliferation and differentiation. They help in proliferation and differentiation. Means if the cell's size increase, okay, and if cells number also increases, it is called as a proliferation. And if they change into their cell types, that is called as the differentiation. Further, the first one is the lymph nodes. Okay, lymph nodes are small solid structures which are present almost everywhere in our body, and one more thing is they help in filtering the lymph. So if any blood-borne antigens do not reach, if it filters the lymph. The next organ is the spleen, which is a bean-shaped organ, and all of you know it is a graveyard of RBC also. It also filters and traps the antigens. And tonsils present in the throat also act as the filters to protect the body from bacteria. And Peyer's patches, see clusters of lymph nodes that are present along the ileum part of the small intestine are called as the Peyer's patches. And Peyer's patches also able to produce lymphocytes, and they also provide sites for reaction between antigen and antibody. And the most important is MALT, mucosa associated lymphoid tissue. So 50% of the total lymphoid tissue is occupied by the MALT, and mucosa of the respiratory, digestive and urinary tract is the MALT. It is present on the walls of respiratory system, digestive tract and the urinary tract. Now we move on to other topic, that is types of immunity. So first we learn about innate immunity. See, inborn immunity is called as innate immunity, means the body possesses immunity by birth itself, that is called as innate immunity. It is non-specific, means this immunity works overall protection it gives. It is not meant for any one type of pathogen or any one type of disease, and it acts by different types of barriers. See students, listen carefully. There is no role of any antibodies in innate immunity, whereas there is role of antibodies in the acquired or adaptive immunity. So what are and how innate immunity brings immunity or protection to the body is by barriers. Okay, physical barriers, physiological barriers, cellular barriers, and cytokine barriers. So the skin, you know, in school also you would have studied stratum corneum. One protective layer is there lining the skin, which acts as the physical barrier for the entry of the pathogen. It does not allow the entry of the pathogens. And mucous membrane present in our body at various places. If any antigen enters, they will trap. So the antigens will entangle in between the mucus, and they will get killed. Then comes physiological barriers. Okay, so acid produced in the stomach will provide highly acidic environment where antigens cannot survive. Even bile, the strong nature of the bile. Then sebum, cerumen means earwax present in the ears is called as a cerumen, also act as physiological barriers. And lysozyme is present in almost all our body fluids except cerebrospinal fluid. CSF does not possess the lysozyme, and even sweat also does not possess the lysozyme. Tears are the places where lysozyme is produced in larger amounts. Then comes the cellular barriers, PMNLs, you know very well. Polymorphonuclear leukocytes means neutrophils, monocytes, macrophages, and NK cells. Macrophages, monocytes, and neutrophils will perform phagocytotic activity so that the antigen cannot attack us. And natural killer cells also, they will punch the cells, they will form the holes with the help of certain proteins called as the perforins. So when holes are punched onto the microorganism, surrounding tissue fluid will enter into it, and so that the cell bursts. Now comes the cytokine barriers. Three types of cytokine barriers are there: interferons, interleukins, and lymphokines. See, cytokine, all these three types function is cell communication and cell signaling. Interferons we have already studied in board classes that, see, these are the glycoproteins produced from the viral infected cells. Okay, viral infected cells. And interleukins also pass the signals to the healthy cells. If any cell, interleukins and lymphokines both pass the signals to the healthy cells if any cell is infected by the virus. Now we learn about the acquired immunity. Acquired immunity is very important for us because lifelong we will rely on this immunity only. Acquired immunity, acquired, the meaning itself is there. Acquired immunity generally develops after on exposure to an antigen, and it is specific. Okay, it is specific for each type of the antigen. And how acquired immunity develops in our body is by production of the T cells, B cells, means T lymphocytes, B lymphocytes, and the antibodies. And here I told you in innate immunity that there is no production of the antibodies. Now let us learn T lymphocytes. See, T lymphocytes are of four types. First type are helper T cells. Okay, so the surface of the helper T cells will have CD4 markers. CD refers to cluster of differentiated molecules. And what is the main function of helper T cells is they stimulate the B cells for the production of the antibodies. Means unless T cells stimulate, B cells cannot produce the antibodies. MNPs means mononuclear phagocytes, and they stimulate the MNPs for phagocytosis. And TC cells, another type of T cells also, they only will stimulate. So these T cells will perform the cytolysis. Cytolysis. The most important lymphocytes in our body are the helper T cells. Now comes cytotoxic T cells. Already we studied they are stimulated by the helper T cells. So cytotoxic T cells also form perforin proteins they release, which punch the holes in the cells so the tissue fluid flows and the diseased cell as well as the microorganism dies. but not causing much harm to the healthy cells. Then comes the memory T cells. So if any antigen attacks our body, memory cells are also released, okay, to recognize the same antigen's subsequent entry. Then comes the suppressor T cells. If the means it suppresses the function of the T cells as well as the B cells if they over exaggerate. Now we are going to learn about types of acquired immunity. See, types of acquired immunity, main feature is it solely depends upon the immunological memory. Solely depends upon immunological memory. It is of two types, active immunity and passive immunity. Okay, so active immunity is of again two types, natural and artificial. See, if any organism attacks our body, after we are relieved from the disease, the immunity that develops in our body is called as natural active immunity. Okay, natural active immunity. And because of the injection because of the injection of the antigen, but not in a natural way, because deliberately antigens are introduced into our body, the immunity that develops is called as artificial active immunity. Okay, so here, because of the antigen, in active immunity you have to remember one thing, attack of the antigen, okay, causes the body to produce antibodies. Okay, so now vaccination is the best example for artificial active immunity. Okay, so for example, live and killed. See, BCG given for bacterial infection is a live vaccine, and Sabin vaccine, polio vaccine, MMR vaccine, which belong to viral, are also live vaccines. Like the TAB typhoid antibacterial vaccine is a killed antigen is given in the vaccine, and SAC vaccine is also for the poliomyelitis. Okay, so now what is the meaning of passive immunity? Let us learn. See, here I told you, because of the entry of antigen, antibodies are produced by the body, but here transfer of antibody takes place. There is no infection, but transfer of antibodies takes place. That is called as the passive immunity. So natural passive means if the antibodies are transferred from mother to fetus or mother to child after his or her birth, that is called as the natural passive immunity, and you have studied IgA in colostrum. Then comes the artificial passive immunity. From immunized individual, if the antibodies are transferred to the non-immunized, it is called as the artificial passive. For example, horse to man. See, ATS, anti-tetanus serum. ATS refers to anti-tetanus serum, which is given to protect against the tetany, Clostridium tetani bacteria. So, those weakened antigens or bacteria are taken and injected into the horse body. So, the serum of the horse body develops antibodies against the Clostridium tetani. Those antibodies are collected, purified, stored and given to the man as vaccine so that he do not suffer from the tetany. So, it is artificial passive. So, what you have to understand is, in active immunity, after the entry of antigen, antibodies are produced. But in passive immunity, there is transfer of antibodies. Now, let us learn about immunity. Two types of immunities are there. One is called as the humoral immunity and second one is called as the cell-mediated immunity. AMI, antibody-mediated immunity. It is cellular, cell-mediated immunity. See, humoral immunity, B lymphocytes play an important role. See, in humoral immunity, what happens is, the circulating antibodies are released by B lymphocytes into the body fluids, which will bring about the immunity. And see, it does not play any role in organ transplantation as well as in treating the cancer or in responding during cancer type of situations. But cell-mediated immunity is developed by the T cells, and which includes all pathogens, and it is mainly involved in during organ transplantation, and it also can provide immunity against the cancers also. And there are two types of immune responses in our body. One is called as the primary immune response and next one is called as the secondary immune response. See, when first time an antigen enters into our body, Okay, so the immunity that the response that we show is very mild, but second time if the same antigen enters into our body, the immune response will be very high, rapid and intense. That is called as the secondary immune response. Now we learn briefly about the structure of the antibody. Antibody structure is given by one scientist named as Rodney Proter. His name is Rodney Proter. Okay, so Y-shaped molecule it is. This is the base, which is also called as Fc end, and these two are called as the Fab ends, also called as antigen binding site. Antigen binding site, and you know, two heavy chains are there, two light chains are there, both are connected by the disulfide bonds, and the base of the heavy or light chain is called as the constant region. C refers to constant region, and the upper part is called as the variable region. So antigen, with the help of antigen binding site, antibodies will stick to the antigens and immobilize them. Okay, now types of antibodies, will have a look. See, gamed, very famous in many books, it is given gamed to remember easily. First one is the IgG. 80% of the antibodies in our body or IgG antibodies produced in the blood, lymph and intestine. So first most abundant they are. Then comes IgA, second most in abundance, 10 to 15%. We have seen sweat, tear, sweat, tears, saliva, mucus and colostrum shows these antibodies. Then IgM is the largest, first formed antibody even in the fetus, blood and lymph, produced from blood and the lymph. Then IgE, less than 0.1% are IgE antibodies which are present on the mast cells, basophils, and they are involved in the allergic reactions. And IgD antibodies are present on the surface of B cells itself, and they are 0.2%. So gamed, you have to remember. Now we will move on to another topic, that is about the allergy. So what is allergy? See, over exaggerated immune response is called as allergy. Another name for allergy is hypersensitivity. It is also called as hypersensitivity. The substances that cause allergy are called as the allergens. Dust, pollen, cotton fibers, feathers, animals' fur, certain types of food, paints, etc. will cause allergy. So symptoms are sneezing, watery eyes, difficulty in the breathing. Now let us see allergic reaction steps. What are the steps involved in the allergic reaction? See, first there will be production of IgE antibodies if any allergen enters into our body. Then IgE antibodies will stick on to the mast cells and basophils. So there is release of the histamine. So this histamine and serotonin, they will dilate the blood vessels. Okay, so that large amount of blood flows through the vessels, and blood fluid means blood and other fluid also leaks out and reaches the tissue spaces, so that there will be decrease in the BP. And if this allergic condition is very severe, it is called as anaphylaxis, where the patients will die. Best example is penicillin. Okay, some patients cannot tolerate the penicillin. All these stages occur, and other examples are hay fever and asthma. Okay, so there is swelling of the tissues in the respiratory tract. They will press the bronchioles so that the persons cannot breathe properly. That is asthma. So asthma comes under allergy. Hay fever resembles fever, means it shows fever, cold symptoms, everything what we discussed in the allergy. That is the hay fever, mainly because of the pollen grains. People suffer from hay fever. Now we move on to autoimmunity. So what is autoimmunity? We have studied. So the first important point about immunity system is they should be able to recognize self from non-self. So in autoimmunity there is failure to recognize the self from non-self. There are many important factors, genetic, environmental factors, and also certain type of drugs play an important role. And during autoimmunity, there will be increase in the number of T helper cells, whereas decrease in the suppressor T cells. Okay, so examples are Addison's disease, Graves' disease, multiple sclerosis, Hashimoto's disease, myasthenia gravis, and rheumatoid arthritis. RF refers to rheumatoid fever. Now let us learn organ transplantation. For the first time, organ transplantation is done by two scientists named as David Hume and Joseph Kelly. Now let us learn what are the organs that can be implanted. Okay, so bone marrow, lungs, liver, heart, cornea, kidneys can be implanted. Then CMI, cell mediated immunity, plays an important role. See, anything that does not belong to our body is treated as antigen by our immunity system. So when these organs are implanted into our body exhibits one type of immunity, that is cell mediated immunity, where the newly implanted organ will be destroyed. Okay, for that immunosuppression is done. You have studied in microbes in human welfare, cyclosporin A. Okay, so now types of transplants we will see: autograft, isograft, allograft and xenograft. See, if person's own body tissue is implanted some other place, it is called as the autograft, and implant between two identical twins is the isograft. And implantation of the organ from one person to another person is called as the allograft. Kidney transplant is the best example for that. Xenograft is implantation of organs from one species of animal to other species. The best example is eyes from the goats are extracted and are implanted into the human beings. Now we learn about the very important AIDS. Okay, some points about the AIDS. Actually, AIDS was detected, listen carefully, AIDS was detected in 1981 only, but the virus HIV was detected in 1983 by Luc Montagnier. See, one scientist named as Robert Gallo has discovered retroviruses. So, then they later understood that HIV virus is one type of the retroviruses, and all of you know every year 1st December is celebrated as the World AIDS Day, and there is gradual destruction of the helper T cells which are mainly responsible for acquired immunity in our body. That is the reason people suffer from symptoms of the AIDS. So what is the transmission? All of us know it, that sexual contact from mother to child, blood transfusion, infected syringes and needles, through this the AIDS virus will be transmitted. So AIDS refers to acquired immunodeficiency syndrome. So how it infects the cells we are going to learn. See, first retrovirus infects the cells. Okay, then its RNA is introduced into the infected cells. Reverse transcription occurs. So viral DNA is produced. This viral DNA undergoes transcription and forms the DNA, means it forms the viral RNA transcription. So viral RNA is produced, and around this viral RNA a protein coat is developed so that new viruses are produced, and these new viruses will infect the healthier cells. Like that, so damage to the helper cells goes on continuously like this. Now, what are the symptoms? Fever, diarrhea, weight loss, bouts of fever and lethargy. And in later stages, AIDS patients will suffer from cancer also. So what are the diagnostic tests? ELISA and Western blot tests are the diagnostic tests. Treatment. See, antiretroviral drugs we call zidovudine, dideoxyinosine are taken to control the AIDS. But AIDS is not curable. NACO, National AIDS, NACO centers are there in every metro city. It is, and also some NGOs will help in bringing awareness about the AIDS disease. Now, one of the important one which NCERT has not given is the ARC, AIDS related complex. ARC refers to AIDS related complex. Before the onset of actual disease symptoms, the persons will suffer from swollen lymph nodes, fever and sweating at night and sudden weight loss, which are the indication of the severe symptoms of the AIDS. That is called as AIDS related complexes. Then SCID refers to severe combined immunodeficiency. It is a genetic disorder, and children will born without the presence of T cells and B cells, and it is called as primary immunodeficiency, whereas AIDS come under secondary immunodeficiency. Please remember, SCID and AIDS are related to immunity system. That's why immediately after immunity I taught about the AIDS. Now children, we are moving on to the other types of diseases, bacterial diseases. Typhoid, which is also called as the enteric fever. Enteron refers to intestine, because in the later stages there are holes formed in the intestine during typhoid, so it is called as the enteric fever, caused by Salmonella typhi. And all of you know, high sustained fever, constipation will be there, and severe weakness, weakness will be there. Then mode of infection, see, contaminated food, water will cause the typhoid. And what is the treatment? TAB vaccine has to be taken. Typhoid antibacterial is the name of the TAB vaccine. It has to be taken, and also there are certain antibiotics like terramycin has to be taken. Then comes pneumonia. Okay, so now what is the mode of transmission is droplets. Okay, so droplets transmission will be there, and using the utensils. Okay, then comes the symptoms. Fever will be there, chills will be there, and there is congestion in lungs. Okay, congestion in lungs will be there, and causative organism, I forgot to write, Diplococcus pneumoniae. Okay, then we can NCERT mentioned nails and lips will become bluish in color. What is the treatment? The best treatment is sulfonamide drugs. Sulfonamide drugs have to be given. Then erythromycin also should be given to treat the pneumonia. Diphtheria. Okay, so diphtheria is also one of the bacterial disease where there is dry adherent membrane. Dry adherent membrane is formed on upper respiratory tract. So it will cause choking, so they cannot breathe properly. And okay, you are understanding. Now it is caused by Corynebacterium diphtheriae. Plague, which can also be called as the bubonic plague, because it causes painful buboes in armpits. And Yersinia pestis. Yersinia pestis is the causative organism, and it is occurs by rats, and one organism, rat flea, Xenopsilla cheopsis. Okay, Xenopsilla cheopsis. Cheopsis is the fly which lives over the rat, and if rats dominate our house, it will release the organism into the food. Now comes the Clostridium tetani, okay, also called as the lockjaw syndrome. Okay, lockjaw syndrome, where paralysis means non-functioning of the muscles takes place with high fever. The death is definite for this disease if vaccine is not taken. Next comes pertussis, Bordetella pertussis, which is called as the whooping cough. Okay, so now we move on to viral diseases. Common cold. Okay, rhinoviruses, and you know, nasal congestion, fever, heaviness in the head are the symptoms of the common cold, and it is said that without taking any tablet or any medicine also, we will get rid of cold within 7 days. Then poliomyelitis. Poliomyelitis is also called as infantile paralysis. There is one nice word in medical terminology, orophycal route. It enters into the body, then so the persons will be paralyzed. It is a permanent condition. Then comes smallpox. Okay, variola virus causes the smallpox, and chickenpox, varicella zoster, and comes the mumps, paramyxovirus, where inflammation and swelling of the parotid glands occurs. Then rubeola. See, measles and rubella rubella is caused by rubella virus only. This is rubeola virus. Skin rash is very common in all this. Are you understanding? Okay. Now comes SARS, severe acute respiratory syndrome caused by coronavirus. Now what we are suffering is another form of corona, and SARS is very common. Every year it occurs, especially in Chinese countries. Severe acute respiratory syndrome. Fungal disease, what we have is ringworm disease. We have in our NCERT ringworm disease, which can also be called as tinea. It is caused by trichophyton. We have seen in lab also trichophyton. Then comes epidermophyton, and then comes the microsporum. Dry scaly lesions in the nails, skin and scalps, and treatment is griseofulvin. We have to take griseofulvin and one miconazole, one tablet we have to, this is a tablet and this is the application, skin cream disease. Okay, so now we move on to protozoan diseases, that is causative organism is Entamoeba histolytica. It releases proteolytic enzymes called as histolysins. So the species name is histolytica. Okay, and mode of infection, actually it is also mainly by contaminated food. The people who have the habit of eating roadside, okay, so they suffer from the amoebiosis. And infective stage. Okay, see, very carefully remember, tetranucleate cyst. Tetranucleate cyst is the infective stage. Where it lives? It lives in the large intestine. Large I means large intestine. And what are the symptoms means? Constipation will be there. Abdominal cramps are there. Abdominal cramps. And then comes the stools with mucus and blood clots. Treatment: mebendazole and albendazole tablets are given. Mebendazole and albendazole. Now comes leishmaniasis. Okay, Leishmania donovani causes this, and Kala Ajar is also it is called. Trypanosoma gambiense causes African sleeping sickness. Trypanosoma gambiense. Okay. Then comes Trichomonas vaginalis. Okay. Malaria. Now let us learn about the malaria. Charles Laveron is the scientist who discovered malaria, and Galgi observed it in the RBC, and Donald Ross observed in the female Anopheles mosquito. Now let us learn what is the infective stage. Okay, see, sporozoite is the infective stage, and asexual life cycle occurs in the man, sexual life cycle in mosquito. So man is called as a secondary host, and this is called as the primary host. Okay, so first sporozoites enter into liver through the blood. They change into cryptozoites. Okay, very soon they enter into RBC. Okay, and here they change ultimately after undergoing certain processes, change into merozoites. Merozoites will release hemojoin granules. These hemojoin granules are actually responsible for the fever. Okay, so for every 48 hours, this whenever hemoglobin granules are released, our body suffers from the fever. Then what are the symptoms that we learn? There are three stages. First one is called as the cold stage, hot stage, and next one is called as the sweating stage. See, during cold, we suffer from severe chills, then the body temperature rises, and after some time it comes down the normal body temperature. There we show profuse sweating. See, quinine, the medicine extracted from Cinchona officinalis is the best treatment. Along with that, paludrin is also one of the medicine, and then comes the chloroquine. Okay, so then comes prophylaxis, means eradication of the mosquito should be done. Okay, that is gambusia, larvicidal fish called as gambusia helps. Then different types are there. Plasmodium vivax we are studying. Plasmodium falciparum is there. Plasmodium falciparum. Plasmodium malariae is there, and Plasmodium ovale is there. So see here, Plasmodium vivax, Plasmodium falciparum, Plasmodium malariae, and Plasmodium ovale. So it causes cerebral malaria, which is very, very dangerous. Plasmodium falciparum causes cerebral malaria, which is very, very dangerous. Now we move on to filariasis. Okay, and you know filariasis, Wuchereria bancrofti, and one more is also there called as the Malai. Digenetic parasites, two hosts are there. Even Plasmodium vivax also digenetic parasite. One is Culex mosquito and another is the man. Okay, it lives in the lower limbs of the body, and in males it lives in the scrotum. So what are the symptoms? Continuous filarial fever, lifelong they suffer, and there is swollen leg which resembles an elephant leg. Then microfilaria larva, here very interesting point is there. Microfilaria larva is the infective stage from man to mosquito. Please remember, don't forget. Okay, then microfilaria larva shows nocturnal periodicity. During night time it comes to peripheral blood vessels. When mosquito bites, it enters into the mosquito. So microfilaria larva transfers from man to mosquito, please remember. And Ascaris lumbricoides, Ascaris lumbricoides, it's a monogenetic parasite, only man, and Ascaris lumbricoides lives in the intestine. Symptoms is internal bleeding, you have studied, that is then comes the muscular pain, then comes abdominal discomfort, and infective stage is one larva is there. Second stage rhabditiform larva is the infective stage. Okay, so now we move on to cancer. Okay, so neoplasia means new growth, neoplasm means tumor or cancer. See, cancer occurs because of the uncontrolled proliferation of the body cells. Cell cycle irregularity leads to the overproduction of the cells, which is called as the cancer. So what are the properties? First, uncontrolled cell division, metastasis. See, when cancer is formed at one place, it is not localized there. A tissue will pinch off from it through the blood circulation. It reaches other organs and causes cancer over there again. That is called as the metastasis. Angiogenesis. See, formation of new blood vessels is called as angiogenesis. Okay, to grab the nourishment from the healthy normal cells, it produces new blood vessels. That process is called as the angiogenesis, and they do not have contact inhibition. See, normal cells, if they come in contact with any body surface or any other cell, they stop dividing so that they will maintain a constant number of cells. But cancer cells have lost this property and they escape from immunity system. Especially NK cells of the immunity system will easily detect the cancer cells, but they will escape from the NK cells. So they grow. Okay. Now two types of tumors first we learn. Benign tumors and malignant tumors. See, that is, benign tumors are harmless and they are localized, means they do not exhibit any metastasis. But malignant tumors or cancerous tumors are harmful and exhibit the metastasis. See, the genes which are responsible to cause cancer are called as the oncogenes. See, when oncogenic transformation of the normal cells occurs, we get the cancer. So, tumor suppressor P53 is the tumor suppressor gene. If any mutations that occur in P53 gene, it changes into tumor promoter gene. P53 controls the cell cycle regulation. If any mutation occurs, it leads to overproduction of the number of cells, which leads to the cancer. Now, what are the causes for cancer? Physical, chemical and biological reasons are there, causes are there. Radiations by sun, then coal tar, mustard gas, cigarette smoke, vinyl chloride, if you are exposed, they act as a chemical carcinogens, and biological carcinogens are the viruses. Then what is the diagnosis? Biopsy. See, the suspected part is taken out, made into thin slices and observed under the microscope. That is the biopsy. In CT scan, X-rays are used to get a three-dimensional image of the internal organs. And in MRI, strong magnetic fields are used by non-ionizing radiations. In NEET, they will ask like this only. You should not get confusion. They will give one of the options as ionizing radiations also. Remember, it is non-ionizing radiations. Now, let us learn. So, treatment. How to treat the cancer? See, radiation therapy, surgery, chemotherapy and immunotherapy. In radiation therapy, X-rays are focused so that no harm occurs to the healthier cells, and removal of the entire part of the part which is affected by cancer is done in surgery. And in chemotherapy, certain drugs are used. So because of this, hair loss and anemia, the people will suffer. And two important drugs are very famous, vinblastine and vincristine. Okay, then immunotherapy by monoclonal antibodies, they will treat it. And one more important textbook has given is alpha interferons, which are called as the biological response modifiers. Now let us learn types of the cancers. See, three types I have made: carcinomas, sarcomas, leukemias. See, every organ or cavity is lined by epithelium. So if the epithelial lining of those organs gets cancer, it is called as the carcinoma. 80% are like that only. Skin, breast, lungs, stomach are carcinomas. Then comes the this one, sarcoma. So mesodermal derivatives when they suffer from cancer, it is sarcoma. Connective tissue and muscular tissue, bones, cartilages, tendons, adipose tissue and lymphomas. Hodgkin's, one type of cancer to the lymphatic system, comes under the sarcomas. And leukemias, abnormal RBC count leads to leukemia. One of the important properties, leukemias are liquid tumors, and World Cancer Day is celebrated on February 4th. Okay. Now, the drugs are classified into three types: opioids, cannabinoids and coca alkaloids. So, from the latex of the plant Papaver somniferum, opioids are taken out, and by snorting or injection, it is taken into the body. The receptors for opioids are present in the CNS, central neural system, and gastrointestinal tract. GA refers to gastrointestinal tract. One of the famous, two are famous: morphine, which acts as a sedative and painkiller after surgery, and heroin is also called as the SMAC. Diacetyl morphine, chemical name, it acts as a depressant, lowers the body functions. Then comes the cannabinoids, cannabis sativa. From the flower tops, leaves, resins, it is taken, and it receptors are present in the cardiovascular system. Marijuana, hashish, charas, ganja are the different types, and its properties are hallucinogenic. Then comes the coca alkaloids, erythroxylum, coca. Okay, cocaine is the main one. It interferes with the transport of the dopamine and causes euphoria and hallucinations. Then LSD, lysergic acid diethylamide, extracted from ergot fungi known as Claviceps purpurea, which is hallucinogenic. And barbiturates are one type of synthetic drugs. Okay, they act as depressant and they induce the sleep. Amphetamines are anti-sleep drugs we call, and benzodiazepines are sedatives. And apart from this, Atropa belladonna and datura are also hallucinogenic. Okay, so now we move on to the strategies in enhancement of food production. This is the important NCERT point. More than 70% of the world livestock population is found in India and China, but it produces only 25%. So animal breeding is of two types, inbreeding and outbreeding. So breeding between the animals of the same breed up to 4 to 6 generations is called as the inbreeding, but continuous inbreeding causes inbreeding depression, and this 2019 this point came what I am going to explain now. So inbreeding exposes harmful recessive genes that are eliminated by selection, most important point, and it also helps in the accumulation of superior genes and elimination of less desirable genes. Now we learn about outbreeding. Okay, it is three types: outcrossing, crossbreeding and interspecific hybridization. See, crossing between the organisms within the same breed up to 4 to 6 generations, but which are not having common ancestors on either side. And outcrossing is the method to treat the inbreeding depression, which is a very important point. And crossbreeding is example is Bicanary eves and Merino rams. Crossbreeding itself tells us that breeding between two different breeds. Okay, so now interspecific hybridization, mule is the best example. Now we learn about controlled breeding even in animals also. One is artificial insemination. Next comes the MOET, multiple ovulation and embryo transfer technology. See, MOET done in cattle, sheep, rabbits and buffaloes. Okay, so first a female cow is administered with FSH like activity. Then because of the injection of the hormones, instead of one egg it produces 6 to 8 eggs. Later that animal is mated with an elite bull or it is artificially inseminated, and fertilized eggs, this is the last point, is very important. Fertilized eggs at 8 to 32 cellid stage are recovered non-surgically, this you have to remember, and transferred to the surrogate mother. Now in fisheries we learn certain points which are very exclusive NEET points I have selected. Examples for freshwater fishes: rohu, katla, clarias, batragus, mrugala, Sirenus mrugala. Marine fishes: hilsa, pomfret, salmon, sardine, mackerel, eel and Bombay duck. In poultry, see in which birds are reared in poultry: chickens, turkey, ducks, geese, pigeons, etc. And poultry birds will suffer from bird flu disease, which is caused by H5N1 virus. Culling means elimination of the sick birds from a group is called as the culling. In bird flu, during bird flu, the birds suffer from respiratory distress, which very soon spreads to other birds. So culling should be done. Now we learn about plant breeding. See, first step is collection of variability, where you have seen in one more question also, germplasm collection plays an important role. It came in NEET many times. The entire collection of the plants or seeds having all diversified alleles for all genes in this one, in crop, is called as germplasm collection. Okay, now evaluation and selection of the parents. Then comes cross hybridization among the selected parents. These are all the steps you have to learn sequentially, one after the other, especially for this one, NEET. Now we learn about Green Revolution initiated by Norman E. Borlaug. Okay, belongs to International Center for Wheat and Maize Improvement, who developed semi-dwarf varieties. Means these plants do not go tall and bend. If they go tall and bend, it touches the water. So the grains will be drenched in water and loss of harm to the farmer. Losses to the farmer. So if the plants are grown at very short height, then, so that it does not touch the water which is logged there, it is beneficial. Those are called as the semi-dwarf varieties. So everything that is given about wheat in NCERT we are going to learn. See, from 1960 to 2000, the production of the wheat increased 11 million tons to 75 million, And hybrid varieties produced in India, semi-dwarf, Sonalika and Kalyan Sona. And other varieties are Lerma Rosa 64A, Sonora 64, Seyful Lerma, Choti Lerma, Sharbati Sonora, and Atlas 66. Atlas 66 is rich in protein and it acts as a donor for many cultivated wheat crops. And insect resistant breeds we have to learn in wheat also. So one type of wheat, wheat is resistant to cereal leaf beetle. Okay. Next one is solid stems of the wheat also does not allow the stem soft fly to infect the wheat. Now we are moving on to rice. Okay, so from 1960 to 2000, the production of the rice increased from 35 million tons to 89.5 million types. And one of the famous semi-dwarf varieties IR8 produced by International Rice Research Institute. And Tai Chung native one is from this one Taiwan, and Jaya and Ratna are Indian varieties. Now sugarcane, two types are there. That is Saccharum barberry, grows in North India, which shows poor sugar content but grows in all types of environmental conditions. Yield is very less. But Saccharum officinarum, mainly in south, it does not grow in North India but shows thick stems and high sugar content. So if a cross is made between these two, the resultant hybrid will have thick stems, grows in all types of environmental conditions with high sugar content. And in hybrids, our NCERT point you have to remember, so such one type of hybrid maize is developed in such a manner that it shows the twice the amount of lysine and tryptophan. Okay, and the low nitrogen and sugar, okay, and high aspartic acid content is developed as an insect resistant protection from the stem borers. Now cotton varieties, if you take, okay, so hairy leaves in the cotton are resistant to jussettes, and smooth leaved and nectarless are to control the bollworms. And BT cotton, you all know, in biotechnology also we are going to study. Then comes the IARI, Indian Agricultural Research Institute, has developed certain nutrient rich vegetables and fruits. In that, for vitamin A content, carrot, pumpkin and spinach, and vitamin C, bitter gourd, batua, tomato and mustard, and calcium and iron rich spinach and batua, and proteins, beans, one type of beans, lab, French and garden peas are developed by IARI to increase the nutritional quality. Now disease resistant crop varieties we have to learn. So first one is the wheat, himgiri and hill bunt and leaf and stripe rust, wheat and himgiri. Next is the brassica, okay, Pusa swarnim. How to remember? This spelling swar, brassica and Pusa swarnim, resistant to white rust. Cauliflower. See, cauliflower we always see for its cleanliness, so like that you can remember the name Pusa shubra is the variety. It appears like a white ball only, so snowball. Black rot and curl blight resistant. Cowpea, cowpea komal, cow, like that you can remember for bacterial blight. And chilies daily we use, so Pusa sada bahar, resistant to chili mosaic virus, tobacco mosaic virus and leaf curl. Now children, insect pest resistant we have to remember. Crop variety is brassica, Pusa gaurav and aphids. See how I will tell you. BG, Pusa is common, BGA. You know BGA, everything related to Facebook. How to remember? I am telling all this. Bad Girl Advice is one of the Facebook page which was abruptly closed in 2019, opened in 2017. Everything about the women it is. But some people are posting objectionable comments and photos, so it was avoided. So Bad Girl Advice. Here B refers to brassica, G is gaurav, A is aphids. Now comes children flat bean, same, JAF. See how you have to remember is, listen here carefully. FB is the Facebook. FB is the Facebook. Okay. And then comes the JAF, just a friend. So Facebook is same like just a friend. Like that you can remember. Facebook is same like just a friend. Or beans is called as same in Hindi. Like that also you can remember. Now comes a okra or bindi. Okay, okra or bindi. Pusa savani. Pusa A4. Shoot and fruit borer. For this also I will tell one B. Then comes the S and A. Here BSA refers to Boys Scouts of America. Boys Scouts of America is the example. So what I want to tell is Boys Scouts of America is established in 1910, which is one of the largest scouts from any country from America all over the world, and its Facebook page also very large. So I have taken this, and here SF, how to remember, San Francisco. Okay, Boys Scouts of America. Okay, America. Four means for San Francisco, like that you remember. Okay, so S and F refers to shoot borer and the fruit borer. Now mutational breeding, what are mutations? See, by applying, see, one scientist named as the Muller is the first scientist to induce mutations. Okay, so even in certain botanical gardens also, one room will be created or constructed. From that, mutation means different types of physical rays, X-rays and other rays are projected, and surrounding that room plants are grown, so that in the plants mutations occur. So Muller for the first time responsible for induced mutations, he applied X-rays on Drosophila. Applied X-rays on the Drosophila. Okay, so actually Muller has done it in Drosophila, but later this technique was used in developing the plants. Okay, so induced mutations means deliberately by applying the physical rays like UV rays, mutations are induced over the plants, so that mutations occur, and whatever desirable character you want, you can develop. So mung bean resistant to yellow mosaic virus is developed, and one more plant is developed that is called as Parbani Kranti. Parbani Kranti. See, Parbani Kranti also most of the books treat it under the mutational breeding, but according to some scientists, see, what is Parbani Kranti? First of all, a type of lady's finger, and whose scientific name is Abelmoschus esculentus. Abelmoschus esculentus. See, according to some scientists, okay, see, from the wild variety and domestic varieties are fused together so that Parbani Kranti is there. But what NCERT is given, that only we have to take under consideration. So once again I want to tell you how to remember Brassica, Pusa, Gaurav and Fs. Listen carefully. In Brassica you take B, bad. Gaurav, okay, it is girl. A, advice. Okay, bad girl advice was very famous in the FB, Facebook. Okay, now it is not there in Facebook because of objectionable photos and messages women are posting. It is all women group only. Now flat bean, Pusa same 2, Pusa same 3, Justids, aphids and the fruit borers. Here again FB refers to Facebook, okay, and JAF refers to just a friend. So nowadays Facebook became a friend only, so like that you remember, Facebook is same as just a friend, like that. Then okra, bhindi, okay, so BSA, I told you BSA refers to Boy Scouts of America. 4 means fur, like that you take fur. SF refers to San Francisco, present in US, and in 1910 it is established. It's a very prestigious institute. So to remember these plants I have used this, Boy Scouts of America. Now children, there is one more type of breeding for enhancement in the food production, that is the polyploidy breeding. One of the famous breeding is Triticum. wheat, and secale cereal, rye, is taken. See, triticum is tetraploid, you can see here, and secale cereal is the diploid. Okay, so if 4n is the chromosomal complement, the germ cells will have 2n, and here we will get n. And 3n type of plant we cannot imagine, because during crossing over or during meiosis there is no proper pairing of the chromosomes. So chromosome doubling will be done by using one chemical. All of you know it is called as the colchicine. When see first once again, triticum is tetraploid, secale cereal is diploid, but the germ cells 2n, this is 2n and this is n. When these two are combined, 3n, triploid is formed. Okay, triploid cannot continue the generation, so chromosome doubling will be done by colchicine treatment. Okay, and triticale became the first man-made crop. Okay, so now we will learn about SCP. Okay, so what is SCP? See, single cell protein. SCP refers to single cell protein. So single cell protein is very nutritious also. It is very nutritious. And see, for example, meat, for the to produce or to extract the meat, we will feed the animal with certain grains. See, 3 to 10 kg grain is required to produce 1 kg of meat from the animals. So it is very costlier process. That's why SCP is done to produce the protein content with less amount of energy, less amount of materials. And in SCP, mainly from cyanobacteria, spirulina is used to extract the SCP. And from bacteria, you know, Methylophilus methylotrophus. If you remember, our NCERT has given 250 grams of Methylophilus methylotrophus can produce us 25 tons of protein because of its high biomass, rate of biomass production. Okay, whereas the 250 kg cow can produce only 200 grams of the protein. Now another is yeast, Candida utilis. From this also SCP can be produced. And potato processing industries will release certain water, and even molasses industries also. All the wastewater that comes out of the industries is captured, and over that organisms are grown to extract the SCP. Now children, we are going to learn about plant tissue culture. See, in this chapter, to enhance the production of the food, okay, and also to limit the workload and to get it in a lesser number of time, different strategies are followed for the nutritional quality of the food and also for high yield. That is the meaning of this chapter actually. If you take animal breeding, increasing the production and disease resistant varieties. Even in plants also, increasing in the production disease resistant varieties we have to develop, because if the plants are not suffered from any diseases, then only they can give high quality food. Even it is same for the animals also. So that is the reason we have developed disease resistant plants. See, for plant tissue culture, 3 are very famous. One is proper preparation of the nutrient medium, providing aseptic conditions, and aeration, proper aeration to the developing tissues. Now listen, see, plant tissue culture is done in laboratory, where the means inside the test tubes. Okay, so these test tubes are placed in different many number of trays, and all these trays are stacked one above the other, so that within a small room you can produce many plantlets of the desired plants. Okay, so nutrient medium, the very famous nutrient medium that is utilized in plant tissue culture is MS medium. MS medium refers to Murashige. Murashige medium is MS medium, where as a carbon source sucrose is added vitamins are added, amino acids, PGRs means plant growth regulators, like different types of plant hormones, auxin and cytokinins, coconut milk added, and in some tomato juice is also added. So this is the perfect nutrient medium in the case of plant tissue culture. First, the test tubes are sterilized, then the nutrient medium is prepared, okay, in the incubator it is grown. Now let us study other things. After adding the nutrient medium, so a cell from the desired plant is taken and grown as the explant. Okay, so what is the meaning of explant? Means a plant cell or tissue that is selected to perform tissue culture is called as the explant. In the test tube, first test tubes are sterilized, to that nutrient medium is added, then explant is dropped over there so that it changes into a white cottony mass. First white cottony mass, that is called as the callus. By absorbing the nutrients from the surrounding medium, it grows and forms the callus. And this explant later changes into plantlet within the test tube itself. Later, these plantlets are slowly taken out of the lab, exposed to the sun for a limited amount of time, say for example half an hour or 1 hour. This process is called as the hardening. Okay, after hardening is done, all these plantlets are grown in the open agricultural fields. Means the trees and shrubs are not formed in the test tubes itself. Only plantlets are formed and plantlets are sown outside. Now what is the meaning of totipotency? The word is given by Haberland. Okay, see, ability to change into many other types of cells is called as the totipotency. And you know what is the meaning of micropropagation? Production of thousands or large number of plants at a time by plant tissue culture method is called as the micropropagation. And one of the NEET questions on this chapter always they ask is meristem culture. And you know meristems are developing tissues of the plant body which are present at the shoot tips, and generally the meristems present at the shoot tips and nodal segments are only selected for meristem culture. And both the apical meristems as well as axillary meristems can be extracted because these are virus free. Now somatic embryo culture. Somatic embryos are prepared by using high concentration of the 2,4-D. Now you see here one point I have written. High concentration of the auxin in plant tissue culture will initiate the roots, formation of the roots, whereas high percentage of cytokinin initiate the formation of the shoots. Now we learn protoplast culture and somatic hybridization. See, for protoplast culture and somatic hybridization, two desired plant cells are taken and fusion of the somatic cells. Here there is no role of gametes only. Fusion of the somatic cells is taken so that a somatic hybrid is formed. First step is removal of the cell walls of the plant by cellulases. You have studied chitinases or pectinases. We can dissolve the cell wall. So now the cell is with a naked protoplasm with cell wall dissolved. These two the protoplasms of these two cells, okay, is fused with the help of one chemical that is called as polyethylene glycol. It is called as the polyethylene glycol, and even electrofusion is also employed for this purpose so that the two cells will fuse, which are called as the somatic hybrid. These somatic hybrids very soon change into callus, and callus changes into new plant. This is protoplast culture and somatic hybridization. See, please remember this point, polyethylene glycol. So, and our book has given tomato into potato they have tried, okay, and it is called as the pomato, but it is not that useful. Now children, we are moving on to microbes in human welfare. So I have planned in such a manner that I have prepared some 13 questions from this. Almost the entire chapter is covered. Because of this time constraint, and already it is done 2 times, every chapter we do very briefly. So curd is rich in. Actually, we know that curd is very much rich in vitamin B12. And why vitamin B12 is required for us? Okay. So because of the vitamin B12, our memory power will increase. Vitamin B12 deficiency not only leads to pernicious anemia but also memory loss. So its high content is vitamin B12 only, but along with that it also shows calcium and phosphorus. So all of this is the correct answer. Then the fermented products of the following: toddy. Okay, what is toddy? Toddy is a traditional South Indian drink. Okay, and it consists of only 6% of alcohol or lesser than that. But if it is fermented for longer time, the alcoholic concentration goes on increasing. Now comes dosa and idli and cheese. Okay, so all of this. Now identify A, B, C and D in the table given. See, biological name is Saccharomyces. Commercial product produced is the ethanol. So the organism name definitely will be yeast. Now here you see it is a bacteria. We have to write its biological name, and it produces butyric acid. So Clostridium butylicum. Okay, butylicum is the answer. Now comes the organism is fungi. Biological name is Trichoderma polysporum, and what is its function? Okay, so now let us learn about the bacteria. Streptococcus bacteria. Commercial products it produces is streptokinase, and it is streptokinase is a clot buster. Streptokinase is a clot buster, and Trichoderma polysporum produces cyclosporin A. All of you know it is immunosuppressant, and streptokinase acts as a clot buster. Okay, now lipases are obtained from, okay, 2 organisms are there. One is Candida lipolytica. Candida lipolytica. Next comes Geotrichum candidum. Next is Geotrichum candidum. Pectinases, one organism is there, Bissoclamus fulva. Bissoclamus fulva. And some scientists say even Aspergillus niger also they will extract the pectinases. Proteases, Aspergillus niger is the example. And what is the function of the pectinases? See, bottled juice and fresh fruit juice, you can see the difference. Fresh fruit juice is having some fibrils dissolved in it, which are actually good for our health. But they use the pectinases to dissolve the nutrient fibers so that the fruit juice appears like a uniform liquid. But you never prefer that. Okay. Next comes the statins. Now we learn about primary treatment. One question is there. During the statins I forgot to tell you. So it is extracted from Monascus purpureus. Extracted from the Monascus purpureus, and blood cholesterol lowering agent. It acts as a blood cholesterol lowering agent. Now the question you see, during the primary treatment in a sewage treatment plant, the solid particles that settle down is called as primary sludge. Listen children, see our textbook has mentioned 2 types of treatment: primary treatment, secondary treatment. There is another treatment which is called as the tertiary treatment, where chemicals are used. In primary treatment, second question is that only, in primary treatment there are 2 processes: filtration and sedimentation. After filtration is over, whatever the particles that are settled at the bottom are called as the primary sludge, and these are extracted and used as manure and also for the production of the biogas. After sedimentation is over, the liquid that is floating on the top is sent to the secondary treatment, and secondary treatment is also called as biological treatment, where all of you know flocks, okay. So fungal filaments with bacteria which are aerobic are called as the flocks. Flocks will act on the water that comes to the secondary treatment, okay. So in sewage treatment plant, the flocks settle in the settling tank. This is called as activated sludge. This is called as the activated sludge. The function of the flocks is to reduce the BOD in the sewage water, so it becomes a pure water, okay. So now 1 minute. Now we learn another question. The technology of biogas production was developed in India by KVIC and IARI, Khadi Village Industries Commission and Indian Agricultural Research Institute. Which of the following can be used as biofertilizers? You can see here, correct option is more than one option. Azotodesmic blue-green algae. Actually, azotodesmic word refers to lichens. See, you know very well that lichens can have algae, pure algae, or blue-green algae, as the phycobiont. Okay, so this blue-green algae, lichens with blue-green algae can also be called as azotodesmic blue-green algae. Okay, and these do not act much as the biofertilizers, whereas nitrogen fixing microbes and mycorrhizal fungi will act as the biofertilizers. And you know very well, mycorrhizal fungi helps in the absorption of the phosphorus, okay, and they provide resistance to the plant for salinity as well as the drought conditions, and nitrogen fixing microbes are different types. For example, listen carefully, bacteria or, okay, now you listen, bacteria and cyanobacteria, means true bacteria and cyanobacteria we take. Here I want to tell you one thing. So for free living nitrogen fixing we will take azotobacter, then comes the azospirillum, and also we can, yeah. Now for cyanobacteria we can take some of the nostoc and anabina. Then comes one of the important point once asked as extra in NEET, Alocera fertilissima, that I will explain later, and these are all free living. If you take symbiotic ones, best example all of you know, Rhizobium in Leguminosae plant family, whereas Frankia in the non-Leguminosae, non-legumes, okay, nitrogen fixer. And here if you take symbiotic, very great example is there, Azolla pinnata, and another organism is Anabina azolla. Listen children, Azolla pinnata is a pteridophyte, Anabina azolla is a blue-green algae. So it's unicellular. Anabina azolla lives in the leaf cavities, of azolla and helps in fixing the nitrogen, okay. And Azolla pinnata also grows in the stagnant water of rice fields, okay. When see, Anabina azolla, by living in the leaf cavities of Azolla pinnata, it provides nitrogen to the plant. When harvesting is over, all the water is dried, Azolla releases the nitrogen into the soil, making it more fertile and nutrient rich, okay. So now we learn trichoderma, okay. It is, read the following statements and identify the incorrect. This is the incorrect one. See, trichoderma is a it lives in the root ecosystems of the plants, but it is not algal member. And dragonflies act as the biocontrol. See, utilization of the chemical fertilizers will cause great harm not only to the environment, to the humus, and also all other organisms related to environment. So, biocontrol agents are used. This is one program, IPM, Integrated Pest Management. In integrated pest management, there is utilization of many organisms which do not cause much harm, and also retaining certain pest as it is, means controlling the number and visit of the pest, means infection of the pest, so that chemical fertilizers are not used. By utilizing living organisms, it can be managed. Dragonflies are the best example under integrated pest management, which are employed to remove mosquitoes. Then comes Bt spores. Bacillus thuringiensis spores are mixed. This is also wrong. Okay. So in water, actually mixed in water and sprayed over to the plants. And baculoviruses attack arthropods only. They attack only arthropods. Okay. Then baculoviruses are also called as nucleopolyhedroviruses, which are species specific, and their effect is broad spectrum. Then comes, what is the meaning of a VAM? Okay, vesicular arbuscular mycorrhiza. Vesicular arbuscular mycorrhizae is called as VAM. Okay, so VAM is example for endomycorrhizae. See children, mycorrhizae is differentiated into ecto and endo. Okay, ectomycorrhizae just cover the outer surface of the roots, whereas endomycorrhizae will penetrate into the cortical cells. After penetrating into cortical cells, they form round vesicles. Okay, round vesicles. These are called as arbuscules. So vesicular arbuscular mycorrhizae. And certain plants called as the sugar cane and maize are called as the smoother crops, because they will get their self protection by releasing certain chemicals. They do not allow the other plants to grow near them. Okay, now Alocera, full name is fertilissima. Alocera fertilissima. It is very important because once asked in NEET, it is very common in Indian rice fields. Very common in Indian rice fields. Acts as a, it belongs to cyanobacteria, blue-green algae. And we know that ladybird beetle helps in eradicating the aphids. Okay, so this and biogas and all you study. So biogas is a mixture of methane, hydrogen sulfide. Okay, so now let us move on to another one. Which of the following alcoholic drinks is without distillation? Wine. See, these three are fermented broth. See, listen carefully. Fermented broth undergoes distillation, so that these type of drinks are prepared, which are called as hard drinks. These are called as the hard drinks. Now, antibiotics, and all of you know very well Alexander Fleming, Florey and Chain were awarded Nobel Prize regarding the world's first antibiotic, Penicillium notatum. Actually, it was discovered by Alexander Fleming, but its high potential nature is studied by Florey and Chain. Okay, so three together were awarded Nobel Prize for this. But one more scientist is credited, Waxman. Okay, Waxman has discovered streptomycin from ray fungi. Ray fungi are nothing but a group called as actinomycetes. Actinomycetes come under the Monera. See, what I want to tell is, Penicillium notatum discovered by Alexander Fleming belongs to purely fungi only. But later on, Waxman is the pioneer person in bringing many types of other antibiotics. He has discovered streptomycin from one bacterial Monniran group called as actinomycetes. Based on his observations, all the antibiotics which end with mycin, all of them belong to actinomycetes. Now, nowadays, because of Waxman work only, varieties of antibiotics are available. Okay. So, now we move on to the ecosystem. Okay. See, the word ecosystem is coined by Tansley. So, first let us learn like this. All of you know very well that there are aquatic ecosystems and terrestrial ecosystems. For aquatic ecosystems, you can take pond ecosystem, lake ecosystem, okay, estuarine ecosystem, marine ecosystem, like that. For terrestrial ecosystem, you can take forest ecosystem, grassland ecosystem, desert ecosystem, etc. See, ecosystem is as big as a forest or ocean, or ecosystem is as small as a small water drop also. Within the small water drop, we can see inconspicuous. Naked eye we cannot see such type of organisms. So micro ecosystem we call it as. Actually, what is an ecosystem? See, all the organisms living in one habitat or one community will exchange materials in between them, and they are also dependent upon the sun and other herbivores for their life, and there will be transfer of energy. All these aspects together come under the ecosystem. So now let us learn what ecosystem components are. See, ecosystem is having a structure as well as a function. You know, if you want to describe any organism, first you will explain about the structure, then you will explain about the function of, for example, organelles only you take. So like that, the structure of the ecosystem is species composition and stratification. Species composition means the many types of species, many different types of species that are living in that particular ecosystem forms the species composition of that ecosystem. Stratification means, for example, in one area, herbs, shrubs, okay, then comes the trees, okay, all of them belong to plants only, but they are arranged at 3 levels as 3 layers. That is called as the stratification, and tropical rainforest will show good stratification of different types of plants like this. Now, what is the functions of ecosystem so that all the organisms will survive? The first one is the decomposition, next one is productivity, third one is energy flow, last one is the nutrient cycling. Okay, so now let us learn one after the other in the next pages. Now, first we learn productivity, then decomposition. See, there are 2 types of productivity, primary productivity and secondary productivity. See, the rate of the production of the food or biomass is called as productivity. Okay, forming the biomass or food is the production. The rate at which the production continues is called as the productivity. So, primary productivity is by the producers. Okay, by the producers. Okay, so after utilizing the solar energy, the total amount of the food or biomass that is synthesized or formed by all plants is called as the productivity, and all plants means primary productivity, all plants come under the producers. There are 2 terms, one is called as the GPP, gross primary productivity, then comes NPP. There is very famous, all of you know very well that NPP is equal to GPP minus R. R refers to respiratory loss. See children, the total amount of the food that is produced by all the plants, all the food is gross primary production, and the plant cannot utilize 100% of the food that it is prepared. Some amount of food it uses for its respiratory activities. After it utilizes some amount of the food for respiratory activities, whatever amount of food that is remaining is called as the net primary productivity. It is related to producers only. And if you want to measure in weight, we will write like this: meter square per year. And here energy, if you want to mention, kilocalories per meter square per year. Okay. Now comes the secondary productivity. See, by herbivores. Okay. See, after eating on NPP, the rate of biomass production by herbivores, which are also called as primary consumers, is secondary productivity. Listen carefully. After the plants use the food for the respiration, the remaining one is called as the NPP. So NPP is available for the next level of organisms called as the herbivores. After eating that, how much amount of biomass they prepare in their body, that settles in the body, that is secondary productivity. So the rate of biomass production by herbivores or primary consumers is called as secondary productivity. Here I want to write one neat point about herbivores or primary consumers. These are called as key industry animals. These are called as the key industry animals because they change the plant matter. They change the plant matter into animal matter. Please remember, change the plant matter into animal matter. Now, based on the productivity of different ecosystems, or how is the productivity in different ecosystems, we are going to learn. See, high productivity ecosystems you can see in the tropical forests, coral reefs and the oceans. Whereas next is average agricultural crops and temperate forest. Okay, and less productivity will be in grassland and savanna. Means compared to above, it is less. Then low level of productivity you can see in the desert. From this also NEET questions will come. Please remember, high productivity, tropical forest, coral reefs and the oceans. But if any question comes in between forest and ocean, you have to tied up. Forest you have to select. Then average is agricultural crops and temperate forest, less from grassland and savannas, and low deserts. Okay children. Now the most important one mark means MCQ, standing crop. The total amount of biomass I need not tell living organisms. Okay, the total amount of biomass present in the organisms of one trophic level comes under the standing crop. And the total number of nutrients, especially inorganic nutrients, present in soil at any given point of time is called as the standing state. This also you have to remember definitely. Now we will move on to decomposition process. Listen children, it is oxygen requiring process. That's why we call it as aerobic process. Means decomposition occurs only under aerobic conditions. Anaerobiosis and extremely low temperature do not favor the decomposition. Now, in decomposition, detritus plays an important role. So, what is the meaning of detritus? The organic remains, that's all. It may be plants or it may be animals, comes under the detritus. Plants, if you take, the old yellow fallen leaves will form the detritus. Organic remains of organisms is the detritus. And there are detritivores. All of you know detritivores. The organisms which feed on the detritus for their food or survival are called as the detritivores. The best example is the earthworm. Best example is earthworm. Okay. So, the entire decomposition process consists of 3 stages. One is fragmentation, next is the leaching, then comes the catabolism, then humification and mineralization. I want to tell one important point. Fragmentation, leaching and catabolism occur in detritus. In the matter they occur, whereas these two processes occur on the soil. Please remember, the first three steps occur in the detritus, the last two steps occur on the soil. What is the ultimate end? What is the result? Ultimate result is the soil is enriched with the humus. Fragmentation means, then leaching, you please remember, all the soluble substances will go into the deeper layers of the soil, that is called as the leaching. Actually, after fragmentation, catabolism should come, then leaching, but how NCERT gives, that only we have to follow. And humification, formation of humus is called as humification, and NEET they will ask like this, what is the nature of it? See, it is a dark, amorphous and colloidal. Very, very important point. Dark, amorphous and colloidal in nature. Because it is colloidal in nature, it acts as a reservoir for nutrients. Acts as a reservoir for the nutrients. Are you understanding? Dark, amorphous, colloidal in nature. Being colloidal, it acts as a reservoir for the nutrients. Then comes the mineralization. As a result of mineralization, carbon dioxide, water and different types of nutrients reach the soil. The ultimate goal is, the ultimate result is the soil is enriched with humus and other nutrients where the plants grow very well. So decomposition is it definitely occurs in any ecosystem. In such enriched soil, once again there is growth of the plants. Okay. Now energy flow we have to learn. So energy flow and trophic levels we have studied. See, trophic refers to food. Many organisms are related to each other by virtue of their food, or means one organism eating on another organism. So the first level, first trophic level is occupied by the primary producers. In nature, phytoplanktons present in the water bodies, grasses, trees and other type of plants acts as the primary producers. And primary producers are eaten by the primary consumers, which we call them as herbivores also. In aquatic ecosystems, there will be zooplanktons. In other areas, grass, grasshopper, cows, deers, rabbits, etc. will act as the primary consumers. So this is the first trophic level. This is the second trophic level. Then primary consumers are eaten by the secondary consumers. Secondary consumers, I think all of you know, can also be called as primary carnivores. They can also be called as primary carnivores, and the level is T3, trophic level 3. And secondary consumers, examples are birds, fishes, wolf, etc. And these are eaten by the tertiary consumers, which can also be called as secondary carnivores, and they can also be called as climax carnivores, which occupy T4, trophic level 4. So, when one organism eats on another organism, the energy is transferred from one level to another level. So, there is unidirectional flow of energy. Very important point. See, in the form of radiation, sunlight, the energy reaches the earth. It is trapped by the producers, then by primary consumers, secondary consumers and tertiary consumers. So the energy flows only in one direction. It cannot get back into the sun. That is the unidirectional flow of energy. And one more thing you have to remember is, at each level of the four above described, at each level only 10% of the food is available for the next level. That is called as the 10% law proposed by Lindemann. Okay children. Now we have to learn food chains. We have seen there is a linear relationship between all the organisms of different trophic levels which form a food chain. That is called as a food chain. So one of the NCERT point I want to write, Grazing food chain is the major conduit. Remember this point. In textbook also you mark major conduit of energy in aquatic ecosystem. Please remember aquatic ecosystem. Whereas detritus food chain, which is called as DFC, this is called as the GFC. Detritus food chain is the major conduit in terrestrial ecosystem. Very important point, it is asked many times. So grazing food chain is the one through which major conduit of energy occurs in aquatic ecosystem, whereas detritus food chain plays an important role in the transfer of energy. Conduit means transfer of energy in terrestrial ecosystem. And in detritus, listen carefully, in DFC, detritus food chain, detritus is the T1, trophic level 1. In grazing food chain, producers or plants occupy the T1 position. Now, see, if all the food chains are interlinked to each other, then its name is called as the food web. So, one example I have written. You can see here, vegetation is eaten by the rabbit, rabbit is eaten by the jackal, jackal is eaten by the wolf, and ultimately wolf is eaten by the tiger. Or vegetation is eaten by the deer and deer is eaten by the tiger. Like that, mouse eats on the vegetation and it can be eaten by the wild cat or it can be eaten by the fox. Ultimately, fox can be eaten by the wolf also, and wolf can be eaten by the tiger, and wild cat can be eaten by the tiger. See, each individual one is one food chain, but all these together crisscross each other and form a food web. Now, we will move on to ecological pyramids. Okay. See, Charles Elton has devised ecological pyramids for the first time, depending upon the biomass production, number of organisms, then energy transfer. Okay. So, that's why ecological pyramids are also called as Eltonian pyramids. They are also called as Eltonian pyramids. Depending on the shape, three types of pyramids are there: upright, then comes the inverted, then comes the spindle shaped pyramid. See children, except in some cases, pyramid of energy is always upright only, but in pyramid of number and biomass, majority times it is upright only, except few cases that we will learn. This is important point. Pyramid of energy is always upright, please remember. And inverted. Parasitic food chain is the best example for inverted pyramid, and pyramid of biomass in the aquatic ecosystem only is also example for inverted pyramid. And the new one you are going to learn is the spindle shaped ecological pyramid. Okay. See here, forest ecosystem is the best example for spindle shaped ecosystem, where you imagine one single tree. Okay, there is one single tree, only one single tree. On the single tree, many herbivorous birds are living. Small birds are living on a single tree, but all of them can be eaten by one large bird called as the hawk. So, it becomes narrow, then broader, and again becomes narrow. Such type of pyramids are called as the spindle shaped pyramids. Best example is the forest ecosystem. Now children, limitations of ecosystem textbook has given. You have to by heart them. They may come under identify the wrong or right statements. See, ecological pyramids assume food chains are simple. Simple food chains actually does not occur in nature, and it has not given any place for the food webs. And a single species may operate at two or more species. Okay, one species is related to many number of food chains at a time, and there is no place given for detritivores and the decomposers. These are some of the limitations. And now the most important one, ecological succession. Okay, so what is the meaning of a seer? What is ecological succession means? Fairly predictable changes in the ecosystem so that successively new type of organisms go on forming. That is ecological succession, a simple definition. Now children, you see, seer refers to a newly formed level of organisms at one particular period is called as a seer. Like that, the seers are formed successively. Seers are formed successively. Serial stages we call them. Pioneer. See, the first formed one, the first formed organisms or seers are the pioneer. The last formed, where it is near equilibrium with the nature, is called as the climax seer or climax organisms. Now we can see here hydrarch. Ecological succession that is occurring in water is hydrarch, and ecological succession that is occurring on the land is called as the xerarch, or dry lands or deserts is called as the xerarch. Actually, xero refers to dry. It may not be always desert only. Are you understanding? So xero refers to dry area. It's not always desert only. So land. Now I have drawn one easiest way to understand. If you remember, our textbook has given one famous point. Both hydric and xeric leads to mesic conditions as the time passes. Are you understanding? As the time passes, both the hydric and xeric leads to the mesic conditions. Now let us see. See, one xeric area is there. Lichens are the pioneer species. Okay. First, lichens will occupy that area, then it is by masses, then annual grass stage, then forms the perennial grass, and then forms the shrub. Okay, then shrub will change into hard woody trees so that it becomes a forest. Once upon a time there was only lichens, and as the time passed by, different serial stages ended with a climax species that is called as the forest. This is the xerarch. Now down you can see, and in NEET they will ask sequential stages of the xerarch. One after the other you have to identify in a successive manner. Now hydric, actually our book has given hydric. See, a water body will be there. First only planktons develop. These are the pioneer species. The next stage is submerged. They change into floating. Then reed swamp stage. Then comes marsh meadows. So as these successive stages are forming, the water level goes on decreasing. Once upon a time it was a broad water basin with large amount of water. As every serial stage is formed, the water level goes on decreasing. Then it is woodland stage, and again it also changes into a forest. Forest represent mesic condition. So both xeric and hydric conditions lead to mesic. And now the water body now changes into is a land with little amount of the water. That is ecological succession. Okay, so occurring on the land as well as the water. Now there are two types of successions: primary succession and secondary succession. See, primary succession means you understood now. Formation of different serial stages of organisms as the time passes is the ecological succession. See, succession occurs in bare rocks and cooled lava and igneous rocks. Okay, whereas secondary succession occurs in a deforested area or abundant agricultural crops. Once upon a time agricultural crop was there, now it is neglected. Abundant agricultural places or areas. Then comes some submerged areas. Because of the water movement, some areas will be submerged. After some time during water, when water reaches that area, all of the organisms will submerge and die. But after water recedes, again the growth starts from the beginning. That is secondary succession. See, primary succession occurs in such an area where there was no presence of even a single living organism right from the beginning of the origin of organisms. Suddenly it becomes a beautiful landscape. That is called as the primary succession. So soil formation only takes. Now the rocks will wither and form the soil. Soil formation only takes very late. After the soil is formed, plants have to be formed. grown, and because plants reach it there, animals also will reach that area. Once upon a a bare area will become a beautiful landscape with all types of organisms. That is primary succession. And primary succession takes thousands of years, because soil formation only takes longer time, then all the organisms have to occupy, so it is very slow. Primary succession is very slow, whereas secondary succession, already soil, everything is formed. For a temporary period organisms became extinct there, but because of the favorable environmental conditions, once again there is formation of serial stages, so it is very fast. Now we move on to nutrient cycles. See, nutrient cycles can also be called as biogeochemical cycles. Bio refers to living organisms, actually geo refers to environment, chemicals refers to nutrients. There is movement of the nutrients from organisms to environment, and nutrients are called as the chemicals. This is called as biogeochemical cycles. And reservoir for this, most of these nutrients is atmosphere only, and carbon cycle, water cycle comes under, carbon cycle and water cycle come under the gaseous cycles, and sedimentary cycle, phosphorus and sulfur come under the sedimentary cycle. And rocks are the reservoirs, mainly rocks are the reservoirs for the phosphorus and sulfur, and atmosphere acts as a reservoir for carbon and the water cycle. So there will be movement of nutrients from, for example, if the plant dies, or animal dies, all the mineral nutrients will be added to the soil, so the soil becomes enriched. In certain soil again there will be, in certain enriched soil again there will be production of the plants, okay, and from there the energy passes to animals, and again animal dies, and again nutrients will reach the soil. Now children, see, phosphorus cycle is most asked in our NEET. Okay, so that's why I have, whatever is in textbook only I have taken. Here you can understand. See, when rock minerals undergo weathering, all the nutrients are added to the soil solution. Okay, and out of that water or fluid that is present in the soil, some amount will run away because of the raining, floods or whatever, and some soil solution is taken by the producers, which are called as the plants. So by taking these nutrients and also with the help of solar energy, producers will accumulate large amount of food, and that food is eaten by the consumers. Okay, and when consumers die, it becomes the detritus. Okay, when consumers die, the remaining organic remains change into detritus, and on detritus action of decomposition occurs, and again the phosphorus changes into soil solution. Means from the detritus phosphorus reaches a soil solution, and by the withering of the rock minerals also phosphorus reaches the soil solution. Whatever form it reaches the soil solution, whichever way it reaches the soil solution, ultimately it is taken by the producers. This is the phosphorus cycle, and definitely you have to remember this cycle. And this is the end of our today B11, and I wish all the best for our volatile students, cordial bile students, and the students who will attend the Sunday classes, and also NLT batches, and also any other persons who are watching this video. And all the best children, all the best. And one paper I am displaying, that is ecological succession, hierarchy and hierarchy. So 3 minutes I will expose that paper. If anyone wants to draw it, and once again I am telling all the best. So please, all of you, if anyone are interested, you please draw the same diagram. It is one of the famous during my degree studies we have learnt like this. Okay, so you take this. 3 minutes I will keep this. I will read out. Lichen stage, mass stage, annual grass, perennial grass, shrub and forest. Climax, it is hydric, plankton, submerged, floating. This textbook has given, so no need. If you want, you draw xeric stages, which will be asked as extra question. And you write like this also. Xeric and hydric both lead to music. As the time passes, climax stage will be reached. Okay, children, all the best.